Okay. First, I'd like to thank Dalvin for this invitation.
Sure.
I think this is I'm not one of the true veterans of this course. I think this is my 4th year, so I still have a way to go to get caught up with the rest. My topic is going to be about pain. Now, chronic and neuropathic pain are certainly, I would call, not an important unmet medical need, even today. The current toolbox of pharmacological treatments leave much to be desired. Treatments are partial, many people don't respond very well at all, and this has prompted a search for better, novel, and more efficacious treatments for this pain. Now, if you look at pain transmission pathways, understanding how this works helps you identify components which one might sort of leverage to use as targets for a therapeutic intervention. Now, since pain nociception is generated in the periphery, a good place to start is with the peripheral nerve. So if we look at the peripheral nerve, just very basic anatomy, which you may know already, it's made up of 3 components. There is the external epineurium, the perineurium, which covers the nerve fascicles, and the innermost endoneurium, which has the fibers which are with without myelin, supported by Schwann cells, fibroblasts, some mast cells, which I've highlighted. The reason for that, I'll come back to mast cells, and microvessels, and also parasites, parasites, in this matrix of collagen. Now, the environment of the endoneurium is controlled by 2 factors. There's a blood-nerve interface made by the microvessels and the covering perineurium. Now, if you look at this cartoon, it shows you these elements. So you have in the endoneurium the nerve fibers with myelin, some blood vessels, there are some fibroblasts, the collagen matrix, and the perineurium which surrounds this. Now, working together, the endoneurium, which should be endoneurium, and the perineurium regulate what goes in and out of the endoneurium. Again, there is this exchange, blood-nerve exchange with the microvessels. There's also a fluid flow which runs from the spinal cord to the central ganglia to the peripheral nerve and creates an proximal distal hydrostatic pressure gradient, sort of an exercise in fluid dynamics, if you will, like a plumber. Now, what can happen if you have a condition of stress or injury or inflammation? These microvessels may become compromised and become more permeable to albumin. So if this happens, albumin flows into the vessels with fluid, and you end up with swelling and edema. And so this drawing shows better if you have the endoneurium with its microcirculation and the outer perineurium. If you have a compromised microcirculation and the endoneurium is swelled, the endoneurium, this compartment, is not very compliant. So if it swells, it really has no space to go, and you end up with compression on the nerve. And this compression can create all sorts of problems. Fiber deformation, local ischemia, irritation, disruption of the microenvironment, and in general, nerve fiber dysfunction, which can lead to problems with nerve conduction, among other things, and also pain. Now, I've taken this other cartoon of the peripheral nerve. There are many of these in the literature. This one in particular has visual players that one in particular, one of the few that I found that actually includes mast cells. So one asks, why are mast cells important in the nerve? Well, because mast cells, if you go back quite a while, discovered in 1878, they are part of the innate immune system produced from the bone marrow as a precursor. Unlike, they're similar to basophils, but unlike basophils, they circulate, and they circulate as immature cells until they reach a tissue destination. When they settle in their tissue, they mature into mast cells. Normally, we associate mast cells with responses to antigen. They have receptors for IgE. They respond to things like pollen if you have allergies. They also participate in wound healing and tissue remodeling. So they have some good functions as well. Now, another feature of mast cells is that you find them in sites in the body which are close to the external environment. So you find them in the skin when you have a mosquito bite, and the site of the bite becomes red and swollen. That's due to release of histamine from mast cells in the skin. If you are allergic to plant pollen, mast cells in the bronchial airways will release histamine, which causes constriction of the airways and an asthmatic reaction. They're also close to blood vessels, and so when they're activated, they can lead to opening of blood vessels. They're also close to nerves. This is a good example of a mast cell, stained for histamine. So they're very rich in histamine. These granules have a variety of agents. I like to think of the mast cell as a jack of all trades immune cell. They have in these granules a variety of preformed agents: histamine, serotonin, heparin, proteases, some cytokines, TNF, before. They have T and B cell lines, so they can interact with T helper cells. They not only have preformed agents, but they also produce them de novo, like cytokines, chemokines, growth factors, radicals, neuropeptides. They're activated by immunoglobulin, IgE, by complement neuropeptides, cytokines, chemokines, extremes of temperature and pressure. Some people who breathe cold air will have an asthmatic reaction to the cold air. That burst of cold air in the lungs can provoke mast cell degranulation. They also interact with nerve cells by way of adhesion molecules, which I don't have on this slide. They will be found along clusters of arterioles. A good example of a mast cell along a blood vessel, which is stained for histamine. So you can see the degranulation. Granules do travel quite a bit of distance. They're also found close to nerves. This is a small nerve fiber, notogram here. They're also close to nerve endings. Another example, in a nerve close to a small blood vessel and a small nerve fiber, notogram here. So if you think of the distance that the granules travel, about 50 micrometers, they can cover quite a bit of space when you granulate your cell. There's quite a large footprint. One mast cell activated can impact quite a large area of tissue. Mast cells are also found in peripheral neuroendoneurium. There are not a great many, but again, because of the area which they cover, very few of them can have quite an impact. There was some discussion about this for a number of years. The mast cells are not easy to visualize, and people have difficulty finding them because normally what you do is stain them for something which is in the granules. As degranulate, they become transparent, and you can't see them. In fact, mast cells, even after fixation of the tissue, will degranulate. If they degranulate, they lose their contents, we don't see them, then people would have said that the nerve doesn't have mast cells, but indeed it does. This is a mast cell in the brain breaking up, degranulating, and again, the granules travel quite some distance. You find them in the brain. They're on the brachial side of the blood-brain barrier. You find them in the meninges. The histamine released in the meninges is probably participating in migraine. The endocrine mast cell degranulation will cause breakdown of the blood-brain barrier. They also participate in stroke, causing blood-brain barrier breakdown. Now, mast cell activation will provoke edema in nervous tissues. An example is a rat model of cerebral ischemia. The mast cell has degranulated and leading to very clearly tissue edema. Again, in brain ischemia, the swelling involves to some extent mast cell activation. If you take this is a rat model in brain ischemia, treat it with cobalamin. Cobalamin blocks mast cell degranulation, and in this case, it also reduces the amount of brain swelling. This compound, 4880, stimulates mast cell degranulation, so in this case, actually makes the brain swelling worse in the ischemia. These animals are engineered to lack mast cells, and those that lack mast cells have less brain swelling in ischemia. If you look in the peripheral nerve and perform an injury, and then section the nerve and look at mast cells close to the injury site, so they section the nerve at different distances from the injury, and look at mast cells, what you appear to see is the paradigm. These are staining for an enzyme to visualize them. And at the site of injury, what you apparently see with time is the loss of mast cells. They appear to disappear. And if you treat them with glycate to block the degranulation, the mast cell numbers remain or actually increase. So this paradigm actually is like a photographic negative. The mast cells appear to disappear because they degranulate and you can't see them. When you block their degranulation with glycate after nerve injury, they are present. So it says they don't degranulate, so their presence is an indication of protection. The important feature of mast cell activation is that when you see them, people think they degranulate, they explode, and they die, which they don't. They will very rapidly release agents. They will also form the genome and release them over time. Furthermore, after injury, they will proliferate. They can also recycle and degranulate again. So they can provoke multiple hits, which is very important. So it's not just a one-shot affair. They do have short-term and long-term effects in the tissue when there is injury. Now, apart from what I mentioned in the peripheral nerve, mast cells participate in pain transmission both at the spinal level and also at the supraspinal level. So if you have a noxious stimulus, the blood vessel in the tissue can release agents which activate mast cells. Mast cells release agents like histamine, serotonin, which will sensitize the peripheral nerve. And this sensitization will transmit the signal to the spinal cord. The spinal cord, the dorsal cord of the spinal cord, there are mast cells in the dura of the spinal cord. They become activated, they close approximately 2 neurons in the spinal cord. These mast cells in the spinal cord release agents which activate spinal cord neurons, which transmit a signal through the brainstem to the thalamus. The thalamus is very rich in mast cells. Mast cells in the thalamus deregulate, release agents which activate neurons in cortical areas, which in the end lead to what we perceive as pain. An interesting observation is that if you take an inch of the peripheral nerve and look in the thalamus, you will see some hours after an actual increase in numbers of mast cells in the thalamus in response to a peripheral nerve injury. So the mast cells work both spinally and supraspinally in transmitting a pain signal from the periphery to the cortex. Now, I've mentioned only mast cells. Of course, the story is a bit more complicated than that. Microglia, which are also derived from the immune system, play a role in pain transmission. They're activated in pain, in nerve, and an interesting feature is that you often find mast cells and microglia in close proximity. And there is good evidence that both cell types will communicate. There are a number of examples, just a few in the interest of time. For example, mast cells have an enzyme called protease activated receptor to PAR2. Now, when mast cells are activated, they release tryptase, which cleaves this enzyme PAR2 on microglia. This activates a pyrinogen receptor, P2X4, which leads to the production of BDNF, brain-derived neurotrophic factor, which, although it's neurotrophic, in this case participates in pain. And as a complement, cytokines released from microglia will upregulate PAR2 on mast cells. So the two, by releasing complementary agents, can activate each other and form essentially a vicious cycle. Chemokines between mast cells and microglia are upregulated, and for example, CXCR4 released by microglia acts as a chemoattractant for mast cells. Complement participates in mast cell interaction. C5a receptor and peptide are released in inflammation. Mast cells don't make C5a, but they have the receptor. And C5a released by microglia is a strong chemoattractant towards mast cells. So both cells involved in inflammation can work together. Now, there's also evidence that mast cells interact with T helper cells in inflammation. By releasing specific antigen to T cells, they will actually polarize the T cell secretory machinery and form what is called an immunological synapse between the mast cell and T cell. That is the story for another presentation. Now, all of this discussion of inflammation, of course, inflammation is bad, we want to combat inflammation. And there is classical pharmacology, but there's also, one can think of other ways to combat inflammation. Now, you might say that in inflammation, the beginning programs the end. That we have, the body produces agents in response to inflammation, which are pro-resolution. So they are induced by inflammation, intended to resolve inflammation. They're called resolvins. Most of these are living in nature. Some are derived from omega-3 fatty acids. They have anti-inflammatory effects. They're upregulated when there's inflammation. So a strategy for inflammation would be to make use of these natural anti-inflammatory mechanisms to combat inflammation. And our interest is in a group of these lipid molecules which are derived from membrane lipids. They're called acetylamides. They're rather simple molecules. They have ethanolamine and fatty acid. A good example is anandamide, which is an agonist of CB1 cannabinoid receptors. Other examples are this one, polyethanolamide, which has ethanolamine and palmitic acid. There are others made of stearic acid, oleic acid, a whole family of these, which are found in the nervous system. They're produced on demand. So if it is an injury or an inflammatory stimulus, they are generated from the tissue. There is a rather long history of them being anti-inflammatory, analgesic, even anticonvulsant. Basically, stress will produce from this precursor with a specific enzyme when there's pain or inflammation. They're released from the tissue, and they have anti-inflammatory effects, neuroprotection, pain relief. There's quite a literature on this. I don't really want no time to go into that. They are degraded by specific enzymes. One is called fatty acid amide hydrolase. It degrades all of these fatty acid amides. Another one is more specific. It degrades the short chain fatty acids. So one can sort of envision a strategy to give these exogenously these anti-inflammatory agents. Another way to do this is to block or modulate the enzymes which degrade them. And you can do that. If you block these enzymes, you will increase the tissue level of these compounds. And by doing so, you can produce an anti-inflammatory effect. And there are different molecules in development which are quite effective in doing this, and they have rather potent anti-inflammatory effects. So, getting close to the end, just a couple of points. So, you influence endometrium by disturbing fluid dynamics and creating inflammation and edema. You can influence nerve conduction and produce pain. Mast cells are important participants in this. They're early responders. And I think that mast cell activation may be a benefit in treating neuropathic pain. There is actually clinical studies which do demonstrate beneficial effects in chronic neuropathic pain. That was ongoing. Just one last slide, sort of afterthought on the subject of neuropathic pain, which I don't think gets much attention. There's chronic neuropathic pain in the elderly. Now, elderly, I'm not sure what you describe as elderly. I've seen some slides where they break this down, they consider elderly as 65 or older, which is kind of worrisome, especially in my case. Anyway, elderly tend to go, it's one of the most frequent reasons they seek medical attention. Now, it doesn't seem that increasing age is a driver of pain, but elderly go to the doctor for pain because of comorbidity, chronic illness, diabetes, falls, disability, and other reasons. Interestingly, mast cells and microglia become more reactive with age in both animals and also in patients. If you look in the endometrium, the mast cells, which conventionally are few, their numbers increase and they become more hyperreactive, which may contribute to alterations in sensory neurons. And of course, mast cells, because because of these proteases, can also injure pain fibers. Microglia as well are far more sensitive with aging, far more sensitive, and also less resistant to regulation. If you take even mast cells or microglia from the brain of a rat, a young rat, or an old rat, the older ones are far more reactive and less responsive to inflammatory agents. So again, treatment of chronic neuropathic pain is difficult in of itself, even more so in the elderly. Just something to think about.